السلام عليكم ورحمة الله وبركاته. طلاب المرحلة الثانية كلية الصيدلة جامعة بابل، محاضرات الكورس الثاني لمادة Human Physiology Endocrine Physiology. Endocrine system is involved in all of the integrative aspects of life, including growth, sex differentiation, metabolism, and adaptation to an ever-changing environment. The endocrine system participates in the regulation of digestion, use and storage of nutrients, growth, development, electrolytes, and water metabolism, in addition to reproductive functions. The functions of endocrine system are closely linked with those of nervous system and immune system. For example, neurotransmitters such as epinephrine can act as a neurotransmitter or as a hormone. The functions of the immune system are also closely linked with those of endocrine system. How? Immune system responds to foreign agents by means of chemical messengers, which called cytokines. Example for cytokines are interleukins and interferons. Also, it can respond to these foreign agents by a complex receptor mechanism. The immune system also extensively regulated by hormones such as adrenal corticosteroid hormone, which is produced by the adrenal gland. What do you mean by hormones? What are hormones? Hormones generally are chemical messengers that are transported in body fluids. They are highly specialized organic molecules produced by endocrine organs that exert their action, the hormones, they exert their action on a specific target cells. So, we need endocrine organ which produce hormones, and we need a specific target cells on which the hormone will exert its action. Hormones do not initiate reactions. Instead, they are modulators of systemic and cellular responses. Most hormones are present in body fluid all the time, but in greater or lesser amounts depending on the needs of the body. A characteristic of hormones is that a single hormone can exert various effects in different tissues, or a single function can be regulated by several hormones. For example, estradiol, which is produced by the ovary, can act on the ovarian follicles to promote their maturation. On the uterus, it can stimulate the growth of the uterus and maintain the cyclic changes in the uterine mucosa. So, estradiol as a single hormone acts on different parts in the body, including the ovary and the uterus. It can also act on the mammary gland to stimulate ductal growth, on the hypothalamic pituitary system to regulate the secretion of gonadotropins and prolactins, and on the general metabolic processes to affect adipose tissue distribution. As you see, estradiol as a single hormone play a major role in different areas and different parts of the female body, including the ovary, the uterus, the mammary glands, the hypothalamic pituitary system, and even on the general metabolism and adipose tissue distribution. We can see in this figure an endocrine cells which produce a peptide hormones. These hormones are present inside the cells in a storage vesicles. When the hormones are released from these vesicles, it will pass through the capillaries from the endocrine cells into the blood, so there will be a diffusion of hormone into the blood. And by this way, the hormone will be distributed to different parts and different areas of the body. We have to learn some specific names in endocrine system. We have the paracrine and the autocrine actions. In the past, hormones were described as chemical substances that were released into the bloodstream and then transported to a distant target site where they exerted their action. Although many hormones travel by these mechanisms, as we said, the hormone released from endocrine cells and it will pass through the bloodstream, transported to a distant target site. By this mechanism, the hormones act. Instead, there is some hormones and hormones-like substances never enter the bloodstream, but instead they act locally in the area in which they are released. These hormones will not pass and transported through the bloodstream. They will act locally in the area where they are released. When they act locally, I mean the hormones, when they act locally on the cells other than those that produce the hormone, the action is called paracrine. When the hormone is released from one cell and exerts its action locally on the cells that are present near the cell that produces the hormone, this action of the hormone is called paracrine. The action of sex steroids on the ovary is a paracrine action. This is an example of the paracrine action, is the action of sex steroids on the ovary. Also, hormones can exert an autocrine action. What do we mean by autocrine action? Hormones can exert an autocrine action on the cells from which they were produced. I mean, the cell that produces this hormone, the hormone will act on the same cell that produces it. This is called autocrine. Example on autocrine action is the release of insulin from the pancreatic beta cells. Insulin produced from the pancreatic beta cells, it can inhibit its release. I mean the release of insulin. Insulin can inhibit the release of itself by acting on the pancreatic beta cell, by acting on the same cell that releases it. This is called an autocrine action. This figure shows us how the paracrine action is performed. As you see, the hormone is produced from one cell. It will release from the cell and will not pass through the bloodstream. It will act on the adjacent cells, so the hormone will diffuse locally and reach to the target cells which are adjacent to the cell that produce it. This is called a paracrine action. And in this figure, we see how the autocrine action is performed. You see one cell that produces a hormone. The hormone will release and act on the same cell that produces it. If we see the figure, we see this tiny, small blue color receptor. So the cell release the hormone, and the hormone have a receptor on the same cell that release it. This action is called the autocrine action. Another form of action is called exocrine action. What we mean by exocrine action? It refers to a mechanism whereby a cytokine that is embedded in or bound to or associated with the plasma membrane of one cell interact with a specific receptor in a juxtaposed cells. What do we mean by cytokines? We have to understand what is the cytokines and how they act. Cytokines are peptides that are secreted by cells into the extracellular fluid and can function as autocrine, paracrine, or even endocrine hormones. They are peptides which are secreted by cells into the extracellular fluid and can act as autocrine, paracrine, or endocrine hormones. Prostaglandins, leukotrienes, and thromboxanes are fatty acid derivatives produced by most body cells. They are produced by most body cells. They are rapidly cleared from the circulation and are thought to act mainly by paracrine and autocrine mechanisms. Their synthesis often is stimulated in response to hormones, and they serve as mediators of hormone action. We have to differentiate between hormones and the mediators of hormone action. The mediators of hormone actions are the cytokines, the prostaglandins, leukotrienes, and the thromboxanes. We have some definitions that we need to understand in endocrine system. What do you mean by exocrine gland? When I said it's exocrine gland, I mean it have a ducts. It is a gland that have ducts that carry their secretory products to the surface, like sweat glands, salivary glands, and mammary glands. While endocrine glands are ductless. So, how the hormone reach to the site of action? Hormones are secreted directly into the capillaries and circulated in the blood throughout the body. While hormone are chemical substances that are synthesized by specific organs or tissue and secreted into the bloodstream and has a specific regulatory effect on the target cell or organ. In the end, what we mean by a target organ? Target organ or tissue are specifically affected by the action of the hormone, and the target organ must have a specific receptors for the hormone. This is make the unique function of the hormone by presence of specific receptors on the target organ or tissue of this hormone. At this step, we have to differentiate the hormones structurally. So we have to classify the hormones according to their structures. Hormones have diverse structures ranging from a single amino acid to complex proteins and lipids. They are usually divided into 4 categories according to their structure. First is the amines and amino acids, including norepinephrine and epinephrine, which are derived from single amino acid tyrosine, and the thyroid hormone, which are derived from 2 iodinated tyrosine amino acid residue. The second type is a peptide, polypeptides, and glycoproteins and proteins. These can be as small as thyrotropin releasing hormone, which contains the 3 amino acids, and as large and complex as growth hormone and follicular stimulating hormone, which have approximately 200 amino acids. While glycoproteins are large peptides hormone associated with a carbohydrates like follicular stimulating hormone. We will have these hormones in the next lectures. Third type of hormones according to the structural classification is the steroids, which are derivatives of cholesterol. The fourth type is fatty acids derivatives, which include eicosanoids and retinoids. This figure shows the characteristics of hormones, and the peptide hormones were on the steroid hormones. So, how hormones synthesize and transpose? According to their structural classification, protein and peptide hormones are water soluble. They are water soluble, synthesized and stored in granules or vesicles in the cytoplasm of the cell that produce these hormones until secretion is required. Usually, when they are secreted, they circulated unbound in the blood. What do we mean by unbound? They are not bounded to any protein. They are free in the blood. While steroid hormone, you have to remember that steroid hormones are derivatives of cholesterol, so they are lipid soluble, released as they are synthesized. What do we mean by released as they are synthesized? They are not stored in granules or vesicles. They are synthesized and directly released from the cells. Example is the steroid hormones and thyroid hormone are carried by specific carrier protein synthesized in the liver. What we mean by a specific carrier protein? That means these hormones are circulated in bounded state in the blood. They are bounded to a specific carrier protein which is synthesized in the liver. Half-life of a hormone mean the time it takes for the body to reduce the concentration of the hormone by 1/2. When the hormone is released, the time that the body takes to reduce its concentration to 1/2 is the half-life of the hormone. Positively correlated with its percentage of protein binding. What do we mean? Thyroxine, which is more than 99% protein bound, has a half-life of 6 days, while aldosterone, which is only 15% bound has a half-life of only 25 minutes. What do we mean by this? Every hormone that released into the blood or into the circulation in a bounded state will have a long half time, like thyroxine hormone, while the hormone that released without bounded state, have no bounded protein, will have a short half time, like aldosterone. In this condition, drugs that compete with a hormone for binding with the transport carrier molecule increase hormone action by increasing the availability of active unbounded hormone. For example, aspirin compete with the thyroid hormone for binding to transport protein. When the drug is administered to a person with excessive level of circulatory thyroid hormone, like a patient with hyperthyroidism, such a drug will cause thyroid crisis, which is a serious effect on the body. If we need to understand more, aspirin as a drug will compete on the binding site with the carrier protein. It will compete on the carrier protein of the thyroid hormone, and thus will leave the thyroid hormone in the circulation for a long time. This will increase the half-life of thyroid hormone. So, how hormone metabolize and eliminated from our body? In some cases, hormones are eliminated in the intact form. Some hormones are enzymatically inactivated at the receptor site in the target cell, where they exert their action. For example, catecholamines, which have a very short half-life, are degraded by catechol or methyl transferase enzyme and monoamine oxidase. In general, peptide hormones also have a short lifespan in the circulation. Their major mechanism of degradation is through binding to cell surface receptors, with subsequent uptake and degradation by enzymes in the cell membrane or inside the cell. While peptide hormones have a short lifespan and are inactivated by enzymes that split peptide bond. Example for this, steroid hormones are bound to protein carrier for transport and are inactive in the bound state. When the hormone is bounded to a carrier protein, it will be inactive. That's what we mean by inactive in the bound state. Their activity depends on the availability of transport carriers. Unbound adrenal and gonadal steroid hormones are conjugated in the liver, which renders them inactive because they are conjugated, they are bound, and then excreted in the bile or urine. This is a form of elimination of steroid hormones. So the liver will help us in the elimination of steroid hormone by inactivation them, and the hormones will be excreted in the bile or in the urine. Thyroid hormones also are transported by carrier molecules or carrier proteins. The free hormone is rendered inactive by the removal of amino acids, which is a condition called deamination. Inactivation of the thyroid hormone is by removal of amino acids in the tissues, and the hormone is conjugated in the liver and eliminated in the bile, the same as the steroid hormones. After we know the classification of the hormones, the structure of them, the metabolism and the elimination, we have to understand the mechanisms of hormone action. Hormones produce their effect through interaction with a high affinity receptors. What do we mean by high affinity receptors? Which in turn are linked to one or more effector system within the cell. These mechanisms involve many of the cell's metabolic activities, ranging from ion transport of the cell surface to stimulation of nuclear transcription of complex molecules. You see how hormones affect all aspects of our life, from the tiny things like ion transport to the important things that we need in our life, including stimulation of nuclear transcription of complex molecules. The rate at which hormones react depends on their mechanism of action. For example, neurotransmitters, which control the opening of ion channels, have a reaction time of milliseconds, while thyroid hormone, which functions in the control of cell metabolism and synthesis of intracellular signaling molecules, require days for its full effect to occur. So some hormones require milliseconds to complete their function, and hormones require days to complete full effects of their function. This is achieved only if there is a high affinity receptor that present in the target cell. Receptors located either on the surface or inside the target cell. The function of these receptors is to recognize a specific hormone and translate the hormonal signal into the cellular response. The structure of these receptors varies in a manner that allows target cell to respond to one hormone and not to another. This is important to specify the hormone action. For example, receptors in the thyroid are specific for thyroid stimulating hormone, and receptors on the gonads respond to gonadotropin hormones only. The response of target cell to the hormone varies with the number of receptors that are present and with the affinity of these receptors for the hormone binding. How? Variety of factors influence the number of receptors that are present on the target cell and their affinity for hormone binding. There are approximately 2,000 to 100,000 hormone receptors molecule per a cell. Approximately 2,000 to 100,000 receptors molecule per a cell. The number of hormone receptors on a cell may be altered for any of several reasons. Antibodies may destroy or block the receptor's protein. Decreased hormone level often produce an increase in receptor numbers. If the cell recognizes that the hormone level decrease, the cell will interact with this reality by increasing number of receptors on the surface by means of process called upregulation. Upregulation represented increased receptor numbers as a reaction to decreased hormone level. This increased the sensitivity of the body to existing hormone level. Likewise, sustained level of excess hormone, if the hormone level increase, often bring about to decreased number of receptors by a condition called downregulation. Downregulation to produce a decrease in hormone sensitivity. So, when the receptor number increase, the hormone sensitivity of the cell will increase. While when the number of receptors decrease, the hormone sensitivity will decrease. The affinity of receptors for binding hormones also affected by number of conditions such as pH of the body fluids, which play an important role in the affinity of insulin receptors. For example, in ketoacidosis, a low pH reduces insulin binding. Ketoacidosis, we mean diabetic ketoacidosis, which is a condition characterized by a low pH. It will reduce insulin binding, and this will affect the condition of the body. As we remember, receptors can be divided into surface receptors, which are present on the surface of the cell, because of their low solubility in the lipid layer of the cell membrane. Peptide hormones and catecholamines cannot cross the cell membrane. As you know, peptide and catecholamines have low lipid solubility, so they cannot cross the cell membrane, which is mainly lipid. Instead, these hormones interact with a surface receptor in a manner that incites the generation of an intracellular signals or messages. The intracellular signal system is termed the second messenger. So, the hormone is considered to be the first messenger. The first messenger is the hormone that binds to the receptor that is present on the surface of the cell. It will interact with the surface receptors, which are termed as the second messenger. For example, the first messenger, glucagon, which is a hormone, binds to the surface receptors on the liver cell to incite glycogen breakdown by way of second messenger system. The most widely distributed second messenger is the cyclic adenosine monophosphate. It is regarded as the most widely distributed second messenger on the surface of the human cells. The CAMB is formed from cellular adenosine triphosphate by enzyme adenylate cyclase, which is a membrane-bound enzyme that is located on the inner aspects of the cell membrane. Adenylate cyclase is functionally coupled to various cell surface receptors by the regulatory actions of G protein. We will take a look of them. A second messenger similar to the cyclic AMP is the cyclic GMP, which is derived from guanine triphosphate. As a result of binding to a specific cell receptor, many peptide hormones incite a series of enzymatic reactions that produce an almost immediate increase in cyclic AMP, which is the most widely distributed second messenger. Some hormones act to decrease cyclic AMP level and have an opposite effect. This figure will give us some cite about the cyclic AMP and the surface receptors. As example of the surface receptors, we see in the first step epinephrine, which is a hormone, bind to the surface receptor, which is called the epinephrine receptor. This binding of epinephrine with its receptor will initiate a series of action inside the cell. This is the adenylase cyclase enzyme. It will lead us to the second step, which is activation of G protein, a transmission of message from the cell surface. This will lead to adenyl cyclase catalyze formation of cyclic AMP. So in this stage we have the cyclic AMP, which is the second messenger. Cyclic AMP will lead the changes inside the cell, which cause activation of protein kinase A, activation of phosphorylase kinase, activation of phosphorylase, and production of glucose from the glycogen. The whole function is terminated with the production of glucose. It began with combination of epinephrine and binding of epinephrine to its receptor, lead to multiple changes that result finally in the production of glucose from glycogen. Sometimes binding of hormone to the surface receptors acts directly rather than through a second messenger. There is no need to cyclic AMP or cyclic GMP. The process will start directly, and there's no need to the second messenger to open the ion channel in the cell membrane. Influx of the ions serve as intracellular signal. The influx of these ions through the ion channels will produce changes in the cell similar to the second messenger changes, and the influx of these ions serve as intracellular signal to convey the hormonal message to the cell interior. In many instances, the activation of hormone receptors results in the opening of calcium channel. The increasing cytoplasmic concentration of calcium may result in a direct activation of calcium-dependent enzymes with their attendant effects. The second type of receptor the second type of receptor, according to their location, is the intracellular receptors. They are not present on the surface of the cell. In this case, hormones like steroid and thyroid hormones are lipid soluble and can pass freely through the cell membrane. They can attach to intracellular receptors and form a hormone superior receptor complex that travel to the cell nucleus. You have to recognize in this stage that the hormone is attached to intracellular receptor from a hormone receptor complex that travel inside the cell and reach to the cell nucleus. The hormone messenger complex then activate or suppress intracellular mechanisms such as gene activity with subsequent production or inhibition of messenger RNA and protein synthesis. For example, as we see in this figure, steroid hormone will bind to the hormone receptor which are present inside this cell. This will lead to a translocation of a steroid receptor complex to the nucleus, binding of this complex to the DNA regulatory sites, transcription and translation through the messenger RNA, and formation of a new protein. The purple part that present inside the cell is the nucleus. The steroid hormone receptor complex pass through the cell and reach to the nucleus, lead to regulation of transcription and translation, and finally a production of a new protein. How the hormone level is controlled. Hormone secretion varies widely during the 24-hour period. Some hormones, such as growth hormone and adrenocorticotropic hormone, have diurnal fluctuations that vary with the sleep-wake cycle. Others, such as female sex hormones, are secreted in a complicated cyclic manner. The level of hormones such as insulin and antidiuretic hormone are regulated by a feedback mechanism that monitors substances such as glucose for insulin and water for the antidiuretic hormone in the body. We will take these hormones in the next lectures, and we will take a look on them. In this figure, we can see three types of hormone regulations can occur in our body. For example, in the first one, a capillary blood contains low concentration of calcium, which will stimulate the parathyroid gland to produce the parathyroid hormone. So the parathyroid hormone will be secreted into the blood. The effect is humoral. It's meant by the blood. In the second type, the preganglionic fibers stimulate adrenal medulla cells. The adrenal medulla cells will produce catecholamines that will secreted into the blood. This stimulation occurs by a neural stimulation. Let's take a look on the third type. The hypothalamus secretes hormones that stimulate the anterior pituitary gland to secrete hormones. This will produce many types of hormones that regulate the thyroid gland, adrenal gland, and the gonadal tissue, stimulate other endocrine glands to secrete hormones. This activation is by hormonal stimulation. So the stimulation may be humoral, neural, or hormonal stimulation. how hormone level is controlled. The first important mechanism for hormonal level control is the negative feedback mechanism, which prevent overactivity of the hormone system. The control variable is often not the secretory rate of the hormone itself, but the degree of activity of the target tissue. What do we mean? In this figure, a simple negative feedback consists of endocrine cell and a target cell. The endocrine cell produces the hormone and cause a positive feedback on the target cell, while the target cell will have a response that have a negative feedback on the endocrine cell. Let's look at a complex negative feedback. The hypothalamus produces hormones that act on the anterior pituitary gland, and the anterior pituitary gland will have a negative feedback on the hypothalamus, while the anterior pituitary gland will produce many types of hormones that will act on the target glands, which will cause a negative feedback on the anterior pituitary and on the hypothalamus itself. So the negative feedback is a complex one. On the other hand, the positive feedback occurs when the biological action of the hormone causes additional secretion of the hormone. What do we mean? When the hormone is secreted and exerts its effects on the target cell, this effect will have a positive feedback on the endocrine glands to secrete more and more hormone. Secretion of luteinizing hormone based on the stimulatory effect of the estrogen before ovulation, while luteinizing hormone stimulates ovaries to produce more estrogen, which is the first stimulatory effect. The first stimulatory effect on the luteinizing hormone is the estrogen. When the luteinizing hormone acts on the ovary, it will produce more estrogen, which will stimulate again the pituitary gland to produce more luteinizing hormone. When the luteinizing hormone reach the appropriate concentration, the negative feedback will occur. There's no need for more luteinizing hormone. Another one is the cyclical variation, the positive feedback, the negative feedback, and the cyclical variation, which is influenced by seasonal changes, stages of the developments and aging, circadian cycle, sleep, etc. For example, growth hormone, which is responsible for development, increases during early period of sleep and will decrease during late stages of sleep. Another example is the gonadal hormones, which is responsible for development of the secondary characteristics and the reproductive function. It will affected by the aging, the seasonal changes, and lunar cycles. The ACTH and glucocorticoids also affected by the circadian cycle. As we see in this figure, which represent our 24 hours hormonal changes, in the midnight, changes will be started. At 2 a.m. we are full in a deepest sleep. At the 4 a.m. the lowest body temperature. At the 6 a.m. sharpest blood pressure rise. At the 7 a.m. melatonin secretion stop because there is a sun rise. At 10 o'clock a highest alertness are present. The fastest reaction at 3 p.m. At 5 p.m., the greatest cardiovascular efficiency and muscle strength. At 6 p.m., a highest blood pressure present. At 7 p.m., the highest body temperature. At 9 p.m., melatonin secretion started again, which will lead us to pass into the deepest sleep at the 2 a.m. This circadian rhythm is affected by our hormones. Another type of regulation of hormone level is the reflex release, which is influenced by stress and new situations. Stress hormones, prolactin, renin, angiotensin, aldosterone systems also affected by reflex release. In the figure we see the breastfeeding have a reflex release of different hormones that results in the production of milk. The level of many of the hormones are regulated by feedback mechanisms that involve the hypothalamic pituitary target cell system. We need here to understand the hormone interactions. Interaction of the hormone is in different ways. First one is synergistic, which means two or more hormones will act together to produce a particular result that is greater than each individual hormone. For example, follicle stimulating hormone and testosterone on the spermatogenesis, cortisol, prolactin, and oxytocin on the mammary gland milk, epinephrine and norepinephrine on the heart. The second type is permissive, which means the presence of one hormone in adequate amount enhance the responsiveness of the target organ to a second hormone. For example, prior to the exposure of the uterus to estrogen, induced formation of progesterone receptors. The third type is antagonistic, which is the action of one hormone will inhibit the effect of the other hormone, like insulin versus glucagon, estrogen, which inhibits prolactin during pregnancy, and progesterone, which inhibits estrogen during pregnancy. The additional value created through synergy and the permissiveness will allow the thyroid hormone, which is no fatty acid release, while epinephrine, small amount of fatty acid release, while if we have the epinephrine and thyroid hormone act together, it will lead to a large amount of fatty acids to released. Insulin versus glucagon. Act each one against the other. Thank you very much.